Green fluorescent protein, or GFP, is a protein which is found naturally in Aquaria victoria jellyfish. GFP, along with its partner protein, aquarion, allows the jellyfish to glow green under certain circumstances. In the early 1990s, scientists were able to isolate and express GFP into other organisms and exhibit its ability to glow without its partner protein or any other enzymes that may be specific to the jellyfish. In the following years, more stable forms and expansive forms of GFP were developed, as well as a wide variety of colours ranging from blue all the way to red. In molecular biology, GFP has been described as a revolution as it allows for the visualization of almost any protein in any cell or tissue. It is used extensively as both a tag and as an indicator. For tagging, the gene of any protein of interest is essentially fused with GFP. This creates a new hybrid protein that not only has all the components and in turn functions of interest, but is also now illuminated. This hybrid protein can now be more easily tracked through and monitored through fluorescence microscopy. As an indicator, GFP are altered through mutagenesis so that they become biochemically sensitive. This allows them to react to certain conditions and environments, which can then be monitored more easily as there is now a change in illumination. Previously, a change in the condition or environment could not be seen visually. For their work in the field of GFP, 3 scientists received the Nobel Prize in Chemistry in 2008. Osuma Shimura for isolating GFP from jellyfish, Martin Califf for expressing it in other organisms, and Roger Tisson for mutating it to create fluorescent proteins in a variety of colours to be used in cells as experimental markers. Although the green fluorescent protein has a scientific history predating 1960, an issue of the journal Science in 1994 is agreed by many to be the beginning of the GFP revolution. The cover image showing green glowing sensory neurons in C. elegans was instantly recognized as representing a breakthrough for cell biology. There are many bioluminescent organisms, but a northern Atlantic jellyfish named Aquaria victoria is one of the latest and more useful to be discovered. It can emit a green fluorescent light which comes from phagocytes along the edge of its body. These lights are generated by an ordered triggering of 2 photoproteins called aquarion and green fluorescent protein. Once calcium binding occurs, the aquarion produces a blue light exciting the GFP, which then takes over, causing the jellyfish to emit a green glow. While GFP in its natural form in Aquaria victoria was discovered back in the 1960s, it was not until the 1990s that the major developments were made, enabling GFP to be used extensively in biochemistry. In 1992, Douglas Prasher led a team that were finally able to clone GFP and determine its nucleotide sequence. This allowed for further research and experimentation, and in 1994, Califf, Inouye and Tsuji were able to express GFP in other organisms. This showed that the illuminating effects of GFP was not reliant on any enzymes that were specific to the Aquaria victoria. It was these major discoveries throughout the early to mid-1990s that opened the door to the use of GFP in biology and across scientific research. Since then, GFP has been researched extensively and improved substantially. Many variations of GFP have been engineered. Some of the major improvements include improved spectral characteristics of GFP, which increases luminescence and photostability, and also the creation of enhanced GFP, which allows GFP to be used in the cells of mammals. Multiple mutations created the superfolder GFP. GFP can still fold and mature, in turn lighting up the protein it is fused with, even when the protein of interest is poorly folding itself. Also, mutations which have led to an array of different colours, including multiple variants of blue, yellow, orange and red, mutations which allow GFP to fold at higher temperatures, and mutations that shift the spectral peak of GFP absorbance and emission peaks. In most cases, a target protein will be tagged by fusion to GFP. However, basic procedures to fluorescently tag RNA are limited. Like proteins, RNA plays many roles in cellular function and is increasingly used for various applications. Thus, further research was required to create a set of GFP equivalents for RNA. Beginning with the derivatives of GFP fluorophore, they then implemented artificial evolution to indicate short RNAs that bind and stimulate the fluorophores. More selections permit spectral tuning of the fluorophores, creating an assortment of spectral properties such as orange, red, yellow, cyan, and assorted greens, in addition to the enhanced GFP like RNA fluorophore called spinach, which allows tagging and tracking of RNAs in living mammalian cells. GFP, as well as its derivatives, have revolutionized the utilization and inspection of proteins for a wide range of uses.
The wild-type GFP found in Aquara victoria protein has 238 amino acids, 3 of which, 65, 66, and 67, transduce the blue chemiluminescence of the protein aquarin into green fluorescent light by energy transfer. Fluorescence taking place upon receiving energy from the calcium-activated photoprotein aquarin. Absorption equaling 395 nanomolar exhibits a smaller absorbance peak of 47 nanomolar. The fluorescence emission spectrum peaks at 507 to 510 nanomolar, with the shoulder at 545 nanomolar. This reactionary transfer of energy allows biologists unlimited access in the study of organic structures, such as the developmental processes of the human fetus. GFP is used as a marker protein due to its ability to attach or mark another protein. The marking of another protein allows biologists to see that particular protein within its organic structure. The GFP gene has also been found to be transmissible, its descendant entities also producing green fluorescence. The detection of GFP only requiring ultraviolet light. It has a special 3 amino acid sequence of serine, tyrosine, and glycine. The serine sometimes replaced by the similar threonine. When the protein chain folds, this short segment is buried deep inside the protein. Then several chemical transformations occur. The glycine forms a chemical bond with the serine, forming a new closed ring, which then spontaneously dehydrates. Finally, over the course of an hour or so, oxygen from the surrounding environment attacks a bond in the tyrosine, forming a new double bond and creating the fluorescent chromophore. Since GFP makes its own chromophore without the need for other cofactors outside of the molecular oxygen, it's perfect for genetic engineering. You simply engineer the cell with the genetic instructions for building the GFP protein, and the GFP folds up by itself and starts to glow. All naturally occurring fluorescent protein chromophores are based on 2 rings linked by a double bond, derived from the dipeptide tyrosine glycine. The minimum energy configuration has the 2 rings coplanar, but both the cis and trans isomers of the central double bond are fluorescent. Within GFP, the chromophore is cis and approximately planar. Applications of GFP are varied. Scientists currently use GFP to tag proteins, cells, tissues, and organelles of interest. In order to interact with or study them, it's used extensively in the study of gene expression, as first succeeded by Professor Mark Chalfie of Columbia University in New York. Having first proven that GFP does not interfere with biological processes, Chalfie then applied GFP to the study of neuron development of C. elegans, having labelled a specific gene with neuron cells with GFP. He was able to observe the amount of fluorescence emitted by the cells. As mutant cells produce less, or in some cases excessive amounts of GFP than normal cells, either amounts would indicate the abnormal development of mutants. Viruses are also able to be tracked within the host by following the fluorescent glow. Just as you would watch through a microscope a protein tagged with GFP moving around in a cell. Scientists are also using GFP as a biochemical indicator and turning them into biosensors, molecular machines that sense the levels of ions or pH and then report the results by fluorescing in characteristic ways. GFP has been very helpful in human gene therapy, drug screening, biological pest control, and observing genetically altered microbes. GFP is an active indicator within the surrounding environment of an organelle. The GFP engineered with potassium channel being the first optical sensor to be genetically encoded in membrane potential. As a reporter gene, it's considered the most powerful monitor of the gene expression, as it does not require any supplement of any additional substrate. Yet considered to be the most thrilling use of GFP is its usage as a fusion tag. The incorporation of GFP can be at either the amino or carboxyl terminus of a protein, and because the fused protein can maintain its normal function along with the GFP expressing gene, all major organelles such as plasma membrane, Golgi apparatus, nucleus, and endoplasmic reticulum can be targeted and observed. An application that is also used is a method allowing serological differentiation between animals that have been vaccinated and animals that have recovered from natural infection. Walsh, Barron, Anderson and Barrett in 2000 discovered that GFP was useful in obtaining a successful humoral antibody reaction to the marker protein. They discovered this by undertaking a trial on selected livestock that they wanted vaccinated against rinderpest, a disease that affects livestock. In order to successfully control and eradicate rinderpest, they produced a genetically marked rinderpest virus (RPV) vaccine. After the trial, they found that all the vaccinated animals were totally protected when tested with the virulent virus.
Over the next few slides, we'll explore some of the key advantages, shortcomings, and future applications of GFP. When comparing GFP to some of the other reported genes and molecular markers, its key advantages as seen on the slide includes heritability, non-invasive and non-toxic, and it only requires molecular oxygen. With heritability, once GFP has been incorporated into the genes of a protein of interest, its luminescent properties will also be passed on to its descendants. Hence, all future descendants of the protein of interest will also exhibit fluorescent properties. This characteristic allows for a variety of applications, including the ability for researchers to not only study the initial interactions of the protein being tagged, but also all the proteins that eventually produce and their subsequent interactions as well. This is currently being applied to cancer research and has allowed for the examination of the cancer cell cycle, mitosis, mortality, evasion, and amongst other things. Clinically, GFP has been used to tag or mark cancer cells. These cancer cells, which now light up when exposed to UV lights, can then be tracked as they spread throughout the body. Further, in cases where tagged cancerous cells are removed, but there is a local reoccurrence, these new cancerous cells will also continue to fluoresce. This allows for easier detection and removal of the new growth. GFP's secondary key advantage is its non-invasive and non-toxic nature. With these properties, GFP can be incorporated into proteins without changing their function. This is done by adding GFP to the carboxyl terminus of a polypeptide, limiting its effect on the overall makeup and in turn the function of the protein being tagged. Additionally, GFP does not affect the cells around it. This enables GFP to be used in vivo testing and allows researchers to examine, monitor, and track these tagged proteins in living cells in real time and within their actual environment. Finally, GFP does not require any cofactors besides molecular oxygen to function. Through its particular and unique cellular makeup, GFP is able to illuminate all on its own. This makes GFP simpler and more straightforward to use. Researchers do not need to worry about how any additional cofactors must be obtained or incorporated, nor do they need to worry about the potential influences that these cofactors may have. The above key features have made GFP immensely useful and popular in biology research over the last 20 to 30 years. However, there are also several shortcomings to GFP. Its key shortcomings include low fluorescent intensity and narrow spectral range, issues with photostability, pH and temperature sensitivity, difficulties obtaining commercial variants of GFP, and interference with protein functionality. So some of these shortcomings relate to the original GFP, which is commonly referred to as wild-type GFP, while others relate to the variants that have been created to combat the initial shortcomings. Finally, some of the shortcomings affect both the wild-type and variant forms. The initial wild-type GFP that was extracted from the jellyfish, while revolutionary, had low fluorescent intensity and a very narrow spectral range. It really only glowed green. There were also issues related to photostability, sensitivity to certain pH ranges, and it could also only operate at around 28 degrees Celsius. To overcome these issues, extensive amounts of research and bioengineering has been conducted over the last 20 to 30 years to create more suitable and diverse variants. For example, there are now multiple colors available, covering almost the entire visible spectrum. This can be seen in the diagram, which shows the wavelength of the most suitable variant from each color group. As we can see, we have variants from 300 all the way to 750 wavelength. There are also variants that are less prone to photobleaching, as well as those that are less sensitive to pH and temperature. This temperature one is key, as these variants can now be incorporated into mammals. While these variants of GFP have greatly increased its application, unfortunately, many of these also have their own drawbacks. For example, there are currently only a few red and orange variants, and those that are available still exhibit poor fluorescent intensity. Further, these newer, more advanced fluorescent proteins can be difficult to obtain, as many are not commercially available. There are also issues around consistency, reported properties, and quality. Finally, issues that affect both the wild type and the newer variants of fluorescent protein includes its large size and its ability to interfere with the function of the target protein. GFP is around 25 kDs in size. In comparison, other organic fluorides are only 1 kD in size. This can make the GFP fused protein quite bulky in size, which can affect the protein's overall structure and in turn possibly its overall function. This is particularly problematic for proteins that are involved in delicate transport processes. So while GFP has been revolutionary, it also has its shortcomings. Therefore, it is pertinent for researchers to select the variant that is most appropriate for their research goals. Finally, this last part of the presentation will discuss some of the future applications of GFP as well as some extensions to the technique. As there are still quite a few shortcomings to the existing fluorescent proteins that are available, research is being conducted to develop variants that are more suitable so that GFP can be applied more extensively and readily. The key areas of interest include the development of a photostable red and orange emitting fluorescent protein, as well as the development or improvement of existing variants so that they are brighter and less susceptible to photobleaching. While some emerging applications of GFP include its uses in oncology and neuroscience. When looking at cancer, GFP is being used both clinically and in research. Clinically, there is currently significant interest around using GFP tagging to illuminate cancerous cells before, during, and after cancer surgery to help differentiate cancerous cells from non-cancerous cells. This allows surgeons to avoid removing healthy cells while reducing the risk of missing cancerous cells. While GFP's non-toxic nature allows for it to be incorporated into the cells of humans, while its ability to light up under UV light allows hospitals to easily utilize it without extensive new equipment. The top image on the left shows some of the ways that GFP is being used in cancer research. Two cells key to cancer have been tagged with different color fluorescent proteins. The macrophage has been tagged with green fluorescent protein, while the tumor tissue has been tagged with a red variant. By using fluorescent microscope imaging, researchers can see how the two cells interact with each other, and in this case, how the macrophage engulfs and digests the tumor cell. GFP is also being used in neuroscience. Here it is helping to illuminate components of the brain that have not been visualized before. This is allowing researchers to better track and observe the interactions occurring in the brain. The bottom image uses the brain bow technique, where each type of neuron in the brain is tagged with a different color fluorescent protein. Once lit up, researchers can see the complexity and intricacies in the brain. Further, it can be applied to study how specific neurons interact with each other and other parts of the nervous system.
In conclusion, GFP has been a revolutionary discovery for molecular biosciences that allows visualization of almost any protein in any cell or tissue. It is described as a marker protein due to its ability to attach or to mark another protein, which allows biologists to see a particular protein within an organic structure, allowing for visualization of molecular processes. GFP comes from a bioluminescent jellyfish named Aquaria victoria, which can emit a green fluorescent light from photokites along the edge of its body. Aquaria victoria is found in the northern Atlantic Ocean. Of the 238 amino acids it has, only 3 transduce into the green fluorescent light. This takes place upon the binding of Ca2 positive, creating an energy which excites the GFP to produce a bright green fluorescent glow. In the early to mid-1990s, major leaps were made in GFP research. In 1992, the nucleotide sequence was determined and GFP was cloned. 2 years later, in 1994, researchers were able to express GFP in other organisms and demonstrate that GFP's illuminating effects does not require any enzyme specific to the Aquaria victoria jellyfish. Then, finally, in 1996, the structure of GFP was determined. Since then, GFP has been researched and improved substantially, with GFP variants, which illuminate brighter, can be used at higher temperatures and in mammals. Further, a range of different colors have also been developed, which has allowed for multiple proteins to be illuminated and observed simultaneously. An example of which is EGFP, which has a brighter green fluorescence and is less damaging to living cells when illuminated. Applications of GFP include gene expression and tagging of proteins, cells, tissues, or organelles in order to study or interact with them. GFP is also used in the tracking of viruses, which is useful in the creation of vaccines.